My name is, oh, sorry. My name is Sylvia. I'm a PhD student at UC Berkeley in Michael Nachman's lab. And today I'm going to talk about the genetic and environmental determinants of adaptive phenotypes in house mice. And the phenotypes that I focus on specifically are complex phenotypes. And the main definition of a complex phenotype is that they're controlled by many loci across the genome in addition to potentially strong environmental influences. And distinguishing between these two influences is important because whether a phenotype is genetically or environmentally determined will greatly impact how the trait arises in a population and how it evolves over time. And for the purposes of what I study, complex phenotypes are often involved in environmental adaptation, with examples such as Allen and Bergman's rule, which are the traits that I focus on. So, this is where animals that are further from the equator have larger bodies as well as shorter extremities, such as ears, limbs, and tails. And these ecogeographic rules are very widespread and they appear across many vertebrate species. And disentangling the influence of genetic and environmental forces in determining these specific complex phenotypes is the main goal of the project that I'll be sharing today. So, that goal is greatly aided by the use of an experimentally tractable system. And that's where the true star of this talk comes in, which is the system that I work with, house mice. So, house mice have a very wide distribution and are found basically everywhere that humans inhabit. But I specifically focus on two inbred lab strains of house mice that were originally collected from Manaus, Brazil, and upstate New York. So these two groups of mice have adapted to very different climates in terms of temperature, with Brazil mice being from a warm tropical environment and New York mice being from a cold temperate environment. And they display classic phenotypic differences that are reflective of this, such as New York mice having increased body size, as well as decreased extremity length. And we think of these phenotypes as thermoregulatory adaptations because they modulate the surface area to volume ratio of an organism. So, large bodies along with small extremities will help retain heat in cold environments, and then vice versa in warm environments. So, getting back to the definition of complex phenotypes that I mentioned earlier, these traits here are undoubtedly influenced by both genetic and environmental factors in wild populations. But these plots here contain data that are from our mice that have been in the lab for many generations under common garden conditions. So we always assumed that the phenotypic variation here reflected solely genetic differences between the mice. However, there is an element of variation that remains unaddressed by these results because these common lab conditions cannot control for any potential non-genetic parental influence on phenotype. And for a placental animal like house mice, we know that the ultimate phenotype, even under common lab conditions, is a combination of the effects of genotype as well as the prenatal and postnatal environment, which is primarily provided by the mother through various actions. And these influences are what we call maternal effects. So, with this idea in mind, I set out to address this large question here of what is the relative contribution of maternal effects and genetic difference to adaptive phenotypes? And further, how are these forces operating to bring about phenotypic difference? So, my talk today will have two main parts. I'll start by testing the presence and action of maternal effects using cross fostering. And then, second, I'll talk about the genetic basis of these phenotypes using QTL mapping. And through using these approaches of environmental manipulation and genetic mapping in tandem, we can, one, determine if phenotypes are genetically or environmentally plastic, or a combination of both. And also, these phenotypes give us an idea, or these methods give us an idea of what the genetic and plastic determinants of the phenotypic variation are. Okay, so starting with part one, to study the presence of maternal effects here, I cross-fostered New York and Brazil litters within 24 hours of birth, and then I measured body weight and tail length every 4 days from birth until weaning. And I also did these measurements for our non-cross-fostered pure line litters. So any difference that we see between the cross-fostered and the non-cross-fostered pups would indicate the presence of a postnatal maternal effect. So I will start with my results from body weight. So here we have New York mice in blue and Brazil mice in orange, and then the cross-fostered pups are in our lighter shades. And what I found is that we do see a strong maternal effect on New York cross-fostered pups early on, where being raised by a smaller mother, that Brazil mother, does lead to less weight gain. But interestingly, this is mitigated once the pups can eat food on their own and not rely solely on the mother's milk. And then ultimately, at 21 days of age, which is weaning, the mice that are raised by their birth mom and the mice that are raised by the mom of the other group are not different weights, which suggests that there's not a lasting maternal effect on weight here. However, when I looked at tail length, I found the opposite to be true. So here we see that New York pups that are raised by a Brazil mother, that light blue, have long tails similar to Brazil mice. And then Brazil pups raised by a New York mother have short tails similar to New York mice, with these groups completely overlapping at 21 days of age. So we can conclude from this that there's a strong maternal effect on tail length, and it's in the direction of the adaptive difference that we see between these mice conforming to Allen's rule. And initially, this was a really perplexing result because we assumed that maternal effects would be acting through mechanisms such as differences in nutritional provisioning that would influence overall growth, not something that would just be influencing tails. So the obvious next question was, what is going on here to make tails so dependent on the maternal environment? And after ruminating for a while, we thought that some previous work in the lab could provide a potential explanation here. So what I'm showing you is the effect of different rearing temperatures on tail length in these Brazil and New York mice. So in this experiment, mice at 3 weeks of age were either kept at room temperature, that's our solid lines, or they were transferred to a cold room, which is the dashed lines, and then their tails were measured every week. And what we can see is that both New York and Brazil mice reared in cold conditions grew shorter tails. So, to test the potential influence of temperature on tail growth in the maternal context, I started measuring the temperatures of New York and Brazil nests throughout the nursing period. So here are the results of those measurements with age of the pups on the x-axis and then our temperature on the y. And what you can see is that nests that are built by a Brazil mother are much higher temperatures throughout nursing, which aligns with that hypothesis I just laid out. So that's all well and good, but the next thing we wanted to think about was what might be leading to this difference in temperature between the nests. And from observing these animals quite a bit, we believe that the most likely cause of this difference in temperature was a difference in nesting, because New York and Brazil mothers do build starkly different nests, with New York having these open and exposed nests, and Brazil having these more enclosed nests. And I used a rubric that ranged from untouched nesting material to having a fully enclosed nest in order to quantify these differences. So what I found is that indeed, Brazil mothers do build more enclosed nests than New York. And we think that this connection between divergent nesting and differences in nest temperature provide us with a potential direct link between the ambient temperature that's controlled by maternal action during rearing and then the ultimate tail length of the organism. So in this last part about environmental manipulation, I want to talk about how the maternal effect is reflected at the skeletal level. And I first wanted to know more generally how the tail length difference between New York and Brazil was achieved. And specifically, I mean, is there a difference in the number of caudal vertebrae? Are the caudal vertebrae themselves different lengths, or is it a combination of those two things? And then secondly, I wanted to know if the components of tail length differed in being genetically versus maternally determined. And I investigated this question using cleared and stained specimen of cross-fostered and pure line mice at 3 weeks of age. So starting with our results from tail vertebrae number, what I found is that Brazil and Brazil cross-fostered mice have about 28 caudal vertebrae, whereas New York and New York cross-fostered mice have about 26. So the lack of difference between the pure line and the cross-fostered mice point to this likely being an evolved genetically determined difference between these mice. And secondly, our results for tail vertebrae length, we have the individual vertebra on our x-axis and then their lengths on the y. And what I found is that our New York cross-fostered and Brazil pups have longer individual vertebrae than the New York mice and the Brazil cross-fostered pups. So this points to this likely being the plastic maternally determined difference here. So through this analysis, I was able to see which skeletal mechanism was conferring the maternal effect. And further, I could determine specifically what elements of the larger phenotype of tail length have a strong genetic basis. And we can take that bit of info into the second part of my talk here, which is focused on genetic mapping. So based on the information that we gained from the environmental manipulation, I focused specifically on the genetic basis of total tail length and tail vertebrae number. And to do that, I used a QTL mapping approach between New York and Brazil mice, which allows you to link genetic variation in a heterogeneous mapping population to phenotypic differences between your starting parental groups. So I will start by talking about our results for total tail length. So here I'm showing you the regions of the genome that showed significant association with tail length divergence between New York and Brazil mice. And there were 7 peaks that rose to significance here. And to make things simpler, I'll just focus in on those right now. And while the confidence intervals for these peaks is a little wide, there are some exciting candidates here that I'd like to share. So under the peak on chromosome 2, there's HOXD13, which has been implicated in tail length differences in many studies. And we know that the overexpression of HOX13 paralogs leads to decreased caudal vertebrae number. And secondly, under the peak on chromosome 4, there's LIN28A, which is another gene involved in tail development. And the overexpression of this gene leads to increased caudal vertebrae number. So going a step further, we can compare our genetic map for total tail length to that of tail vertebrae number to get a more fine-scaled view on the genetic control of this phenotype. And what I found is that 3 peaks rose to significance for vertebrae number, and 2 were shared with total tail length, 1 where that candidate on chromosome 4 is. So this comparison allowed us to get a better understanding of which QTL peaks of our more composite trait of total tail length may be due to a difference in caudal vertebrae number, and which may be due to another element of tail development. So to wrap up, I started by wanting to disentangle the environmental and genetic determinants of complex phenotypes here. And through the combined use of environmental manipulation and genetic mapping, I found that there's a maternal effect on tail length in these house mice that's mediated by a nesting-dependent difference in rearing temperature. And secondly, that tail vertebrae number is strongly genetic and shares a QTL peak with total tail length on chromosome 4. So ultimately, through this work, we're able to describe a previously unappreciated source of variation that's influencing adaptive variation in our system. And it's related to this widespread pattern of longer extremities in warmer environments, which is a pattern that we see across many vertebrate species. So lastly, I hope these results highlight the importance of investigating multiple levels of biological influence in order to truly understand how adaptive phenotypes are generated and maintained in populations. So I think I'm butting up against time, so I'll just say thank you, of course, to my lab and the organizers of this, and to all of you for listening. Thank you.
Great. If anybody has a quick question for Sylvia while we, the next speaker can share their slides.
Please raise your hand if you have a question, then we can ask it.
Okay, let's keep on keeping on. Next is Rosalind Price Waldman. Go ahead, take it away.
Cool, thanks for coming. I'm Rosalind, and I'm really excited to tell you about feather microstructures in colorful birds. So birds are extremely colorful animals, and they produce this color diversity using both pigments and structural colors. Melanin pigments produce browns and blacks, carotenoid pigments produce yellow, orange, and red, And structural colors produce most of the ultraviolet and blue colors in birds, but they can also combine with pigments to produce colors like green. Today, I'm going to be talking about how the microscale branching structure of feathers interacts with these pigments or nanostructures within the feathers to enhance or otherwise modulate coloration. So feathers are made up of tiny branches. The main rachis has small branching barbs and even smaller branching barbules that lock together to form the vein of the feather. This full branching structure appeared fairly early in feather evolution, and it has a lot of functionality in flight, water repellency, and thermoregulation. So when I talk about feather microstructures, what I'm really referring to is these branches, the barbs and the barbules. And then when I say modified microstructures, what I mean is modifications from this ancestral branching structure. Okay, so in the last few years, there have been some interesting descriptions of how modifications in feather microstructures can change the appearance of colors in lots of interesting ways. So for example, in A, the evolution of these curved boomerang-shaped barbules actually enhances the angle dependence of the underlying green structural color, so that a single iridescent feather can produce colors that range from green to orange. And in B, the evolution of these branched barbules in black feathers enhances light absorption, resulting in super black plumage. So these examples are super fascinating, but in general, most of the descriptions of microstructures and color production have looked at pretty narrow evolutionary scales or focused on the interactions of microstructures with a single underlying color production mechanism. So it's really unclear whether these modified feather microstructures are isolated evolutionary novelties or whether modifications to the branching structure of feathers is really a general feature of color production. So in this study, we wanted to understand how important microstructures really are for color production across color space, including in both pigmented and structurally colored feathers. So this work is based in the tanager genus Tangara, which includes 27 songbird species distributed throughout South America, Central America, and the Caribbean. And they have really enormous pigmentary and non-iridescent structural color diversity, which made them a really ideal group for this study. My talk today is based on 4 years of work with a really amazing team of collaborators at Princeton, the Natural History Museum of Los Angeles, the Alexander von Humboldt Institute in Colombia, and the University of Southern California. So first, I'll tell you about how we use museum specimens or feathers from museum specimens to show that microstructural modifications are widespread and enhance plumage brightness and saturation. And then I'm briefly going to tell you about how we use developing feathers that we sampled from wild molting tanagers to study the developmental basis of some of these microstructural modifications. So in the first phase of this project, we really just wanted to know how much microstructures varied in different kinds of feathers and what effects modifications in microstructures had on plumage color. So to answer this, I sampled feathers from 26 Tangara species from museum specimens, and I took feathers from 7 standard patches across the body for each specimen. And then for each of these patches, I collected a range of data to characterize the microstructures and ultimately relate it back to feather color. So I first measured color of the patch using UV-Vis reflectance spectrophotometry. Then I took a single feather from each patch, and I imaged the dorsal and ventral features using environmental scanning electron microscopy. I also imaged the feathers using bright field confocal microscopy, which was a way of getting sort of high resolution images of the 2D features of the feathers in color. One other really important piece was understanding the shape of the barb and how pigments and nanostructures are actually arranged within it. So I cross-sectioned the barbs and then I imaged them in the confocal. So here you can see how this yellowish carotenoid pigment is layered on top of these blue reflecting nanostructures to produce the green color of the feather. And then I finally imaged these cross-sections in the SEM to really look at the nanostructures and make sure that I understood the physical basis of color production in each feather. So in the end, what I had was a really big data set capturing 2D and 3D elements of microstructure. And I also had color data for the patches, which I summarized in terms of brightness and saturation. So I took all these data and I analyzed them using Bayesian phylogenetic multi-level models. So the first takeaway is that there's a ton of variation among different kinds of feathers. So here I'm showing you a confocal image of a melanin-containing feather, and you can see it has these really narrow barbs with relatively long, thick, and dense barbules. But on the other hand, both carotenoid pigmented and structurally colored feathers tend to have wide barbs, and the barbules are really reduced or even absent. So the feathers that I'm showing you here are sort of on the extreme end of this, of having wide barbs and reduced barbules, but there's also a lot of interesting variation in the middle. And in addition to this variation in barbule density and barb width, there are also three-dimensional changes in barb aspect ratio and shape. So here I'm plotting a bunch of these cross sections ranging from structurally colored barbs, which are sort of like flattened and wide, to pigmented barbs, which are sort of teardrop shaped. So the barbs in the middle column have both pigments and nanostructures, and they have these sort of intermediates, but also pretty variable morphologies. And I, was honestly kind of stunned when I started looking at these cross sections, because this is an enormous amount of variation in the shape of these feather branches based on whether they're structurally colored or pigmented. And it suggests that there must be a clear optical function to modifying barbs in these ways. So putting all of this variation together, here's a morphospace that captures changes in barbule density, barb shape, and barb size. The shape of the data here is really striking, and you can see that different colors of feathers occupy pretty different parts of this morphospace. But there's also a lot of interesting continuous variation among feathers, especially feathers that have both nanostructures and pigments, like many green feathers. So taking a step back, what does all of this variation in microstructure actually mean for coloration? So to answer that question, I analyzed these morphological data and our spectral data using these Bayesian phylogenetic multi-level models. And the takeaways were pretty clear and consistent. So first, we found that carotenoid feathers with lower barbule density and narrower, larger barbs are more saturated, because these changes increase the path length of light through the pigmented barb and increase light-to-pigment interactions. Meanwhile, structurally colored barbs with lower barbule density and flatter, larger barbs are brighter, because they're actually maximizing the surface area of the barb that contains light-scattering nanostructures. So again, taking a step back, this means that carotenoid pigmented and structurally colored feathers have actually evolved some pretty substantial modifications to enhance coloration. And a key modification in both kinds of feathers is the reduction or complete loss of barbules, the tiniest branch of the feather. You might remember from earlier that barbules are really important for flight and for water repellency, so it's pretty remarkable that they're absent in the brightest and most saturated feathers. And because this barbule loss is so striking and important, we really wanted to dig into this deeper. So in the last bit of this talk, I'm going to tell you about one really fascinating twist to this story that I personally love, which is that barbule loss appears to happen through different developmental mechanisms in carotenoid pigmented and structurally colored feathers. So if you look closely at a carotenoid pigmented feather, you can see it lacks barbules completely. So here I'm showing you the dorsal surface, close up of the ventral side of the feather, and a cross section. The barb is just like this very thick, smooth structure with no evidence of barbules. But on the other hand, it turns out that structurally colored feathers do still have barbules, but they're actually very small and hidden on the ventral side of the feather. So on the left is a dorsal view of the feather, and you can see these wide blue barbs without visible barbules. In the middle is a ventral close-up, and on the right is a cross-section. And in both, you can see these tiny little barbules, and they're sort of like curled up tightly against the ventral surface of the barb. So this suggests that structurally colored feathers are actually doing something entirely different than carotenoid feathers developmentally to hide their barbules, and that these two types of feathers have independently evolved modifications to enhance feather color via alternative developmental mechanisms. So this is a really interesting example of convergence, but we also all just thought the ventral barbules were really weird and cool and sort of mysterious from a developmental perspective. So in the last few minutes, I'll tell you just a tiny bit about the work that we're doing to understand feather development. So typically feather development is studied by plucking and regrowing feathers from captive birds. That wasn't really feasible with tanagers. So my collaborators, Allison and Gustavo, and I spent a few weeks misnetting molting tanagers in Colombia and sampling their developing pin feathers alongside a really amazing team of researchers from the Humboldt Institute. So I want to emphasize that this was sort of uncharted. This kind of work hasn't actually been done with feathers from wild birds before, and we honestly didn't know if it would work. But we got really lucky and got a good diversity of pin feathers from Tangara and closely related tanagers. And then we collaborated with Chengming Chang and Ping Wu, who are amazing feather developmental biologists at USC. And we used histological approaches to compare feather development among structurally colored and pigmented feathers. And just really briefly, the approach here is that feathers grow from the base, so the tip is actually older than the base. And by taking cross sections at different points along a pin feather, you can actually build a time series of the major developmental events, which we did for a range of feathers with different microstructural morphologies. And again, this wasn't something we were sure would work with feathers from wild birds, and it was a bit of an emotional roller coaster, but the cross sections actually came out really beautifully. So I'm excited to show you some of those data now. So here's a cross section of a developing feather. This is a pigmented feather with full branching barbules that we used as a control, and it's pretty late in development. So these images can be a little overwhelming if you're not used to staring at feather follicles, but the follicle is organized into units called barb ridges, each of which has one barb with paired rows of barbule cells on either side, which I've outlined here. So this is just the tiniest snapshot of the data, but we use these kinds of images to build our developmental time sequences, and then we compared those among different kinds of feathers, including various kinds of controls, to map out what's physically happening in the feather follicle to produce these different microstructural configurations. And to cut right to the chase, we are indeed finding that barbule reduction or loss happens at different developmental stages and involves different parts of the barb ridge in structurally colored and pigmented feathers, confirming that a similar strategy to enhance coloration by getting rid of barbules evolves via alternative developmental mechanisms. And this is not really a surprise, right? Like, even without doing any developmental work at all, it's pretty evident that one kind of feathers lacks barbules and the other kind has ventrally hidden barbules, so they must be doing something different. But by taking this developmental approach, we can start to identify exactly how existing, and in some cases ancient, developmental pathways that control feather morphogenesis and branching can be modified to produce new morphologies. I want to quickly mention a couple of future directions we're working on. So first, we're using immunostaining to map the protein expression of known feather development genes at different developmental stages. And second, we've also looked at microstructures in colorful feathers across birds and have identified a lot of convergence in microstructures, including barbule loss, and also some intriguing evidence suggesting that the underlying mechanisms for barbule loss are even more diverse outside of tanagers. So if you're curious about these other directions, I would love to chat more. So overall, what we found is that variation in feather microstructures is widespread and enhances plumage brightness and saturation. And both structurally colored and pigmented feathers have lost barbules in really different ways. This means that producing bright and saturated coloration often involves substantial modifications to the branching structure of feathers, which I think means that characterizing these microstructures broadly is really important if we want to understand the costs and constraints associated with the evolution of colorful signals in birds. So for example, selection for bright and saturated signals that results in barbule loss likely trades off with selection for other feather functions like flight and water repellency, where barbules play key roles. And by more fully characterizing and appreciating these trade-offs, we can have some new insights into major questions in color evolution, like the origins of sexual dichromatism or latitudinal or elevational gradients in coloration. With that, I want to thank my really fantastic collaborators, the museum curators and staff who have pretty generously allowed me to sample so many feathers for this project, people who have taught me to use a lot of different microscopes, and then my funding sources, especially a grant from SSE. And thank you all for listening. I'm really happy to take any questions.
Fantastic. Put your questions in the chat. Meanwhile, Emily, you want to go ahead and share your slides? Thank you.
Carol, to address your question, do you see variation in microstructure and pigmented barbules across individuals within a species? Yeah, so there are some pretty interesting differences in males and females, and then there are also some pretty interesting differences across the body. The shortest answer is that you often see barbules retained in like the belly or other patches that are really important for thermoregulation, and they tend to be most modified in the crown and other patches that are important for signaling.
Maybe just briefly, if you put your questions in the Q&A section, that might be a little bit better. I think it's available in the recording later on. And I think it's a bit better in terms of overview.
Okay, Thank you. Next up, we have Emily Troyer. Take it away, Emily.
Great, thank you. So I'm really excited today to present some of the work that I've done for my PhD on innovation and morphological diversity in fishes. So evolutionary innovations have shaped the process of evolution and can account for the success of numerous clades. Innovations are these novel traits which directly enhance diversification by allowing species access to these previously unobtainable ecological niches. Examples include things like flight in birds, adhesive toe pads in arboreal lizards, and pharyngeal jaws in cichlids and a lot of other fishes. However, the process in which these innovations arise and their downstream effects on patterns of morphological evolution remain unclear. Modularity and integration have both been proposed as these factors facilitating the evolution of innovation. Traits that are highly modular will evolve semi-autonomously, allowing organisms to evolve these independent adaptations. Alternatively, more integrated traits are evolving together in a coordinated way, and strong trait integration has been associated with really rapid rates of shape evolution and extremely high morphological disparity in a lot of different clades, which leads to the evolution of these really divergent morphological structures, like the asymmetrically flattened skulls in flatfishes and the beaks in these honeycreeper birds. This seems to suggest that integration may be promoting the evolution of these more morphologically disparate phenotypes. Additionally, innovations may also enable access into new habitats, resulting in increased lineage and morphological diversification rates. Coral reefs are these really exceptionally diverse habitats which contain a host of complex interactions between species. And there are numerous reef-associated species that have these novel innovations. One example here is in the butterfly fishes, where they possess this intramandibular jaw joint, which expands their gape size in order to assist on grazing off of reef surfaces. And another well-known innovation is the avian beak, which is a feature that's permitted birds to radiate into these novel adaptive zones, facilitating speciation and increased diversity. But although birds do represent the most speciose clade possessing beaks, they can be found in a lot of other groups as well, including turtles, extinct pterosaurs, and also certain groups of fishes, believe it or not. So among fish, the beak has evolved independently in at least 2 groups, parrotfishes and certain tetradontiform fishes like pufferfishes. Now this brings me to tetradontiform fishes, which if you are not familiar with them, they are perhaps the most wacky group. They represent an incredibly excellent system to explore the mechanisms relating to the evolution of innovation. These guys occupy a wide variety of habitats like coral reefs, freshwater rivers, open oceans, and they include fishes like the porcupine fish, the mola mola, the box fishes, and a lot more. So they also have some really odd morphologies, including these erectable body spines in the porcupine pufferfish, this cube-shaped body in the boxfish, and most interesting to me is the fusion of the teeth into a beak in several of the families. So this beak represents an innovation which has enabled tetradontiforms to occupy novel dietary niches. Beaked species will possess this pair of very hard tooth plates, which aid them in crushing very hard-bodied prey, such as crabs, clams, and other bivalves. So here, using tetradontiform fishes as our model, we wanted to investigate the factors that promote the evolution of innovation. The tetradontiform beak represents this extremely divergent morphological structure, and thus we may expect that the skulls of beaked species will exhibit higher levels of integration and also evolve more rapidly and be more morphologically disparate compared to skulls of non-beaked species. Additionally, highly diverse habitats such as coral reefs may have actually led to the evolution of the beak and may be promoting increased rates of diversification and morphological evolution. So to answer these questions, we use three-dimensional geometric morphometric data from CT scans of museum specimens to quantify skull morphology. We obtained morphological data for 176 species of tetradoniform fishes. And this includes 3 fossil specimens, which are some of the only known three-dimensionally preserved tetradoniform skulls available in museum collections. And these 3D skulls are especially useful for our analyses because if you don't know, most fossils, most specimens, when they are fossilized, they are flattened, and this really complicates the placement of three-dimensional landmarks. So of our 176 species, we generated new data for 145 species, and then we used specimens from Morphosource to increase our taxonomic sampling. After segmentation and digitization in the 3D slicers program, we performed what's called general Procrustes analyses just to remove the effects of any non-shape variation, like the scale, the size, or the orientation of each specimen. We used our previously estimated time-calibrated tetradoniforms tree from Troyer et al., which this includes all species for which we have morphological data. And then each species in our analysis was coded by dentition type and by habitat type. So we coded each species as being beaked if they possessed these highly modified and fused teeth. And then we coded species as non-beaked if their teeth were in discrete units protruding from the jaw socket. And for habitat type, we determined if each species was either reef-associated or non-reef associated based on information found on the FishBase platform. And then for each trait, we reconstructed ancestral states using PhyTools. We then quantified rates of skull shape evolution and morphological disparity using functions in the geomorph package. And lastly, to examine rate shifts across the phylogeny on a specific branch-by-branch basis, we used Bayes traits with a variable rates model. And finally, to look at patterns of modularity and integration between our beaked and our non-beaked skulls, we defined 8 different skull modularity hypotheses, which encompass a range of different functional, embryological, and sensory hypotheses based off of the literature. So then we can take these 8 hypotheses and do model testing with functions found in the geomorph package. And then under our best fit model, we can then compare the levels of skull modularity and integration between our beaked and our non-beaked species. All right, so what we find is that tetradoniforms display this enormous diversity of skull shapes. So in this phylomorphospace plot, each dot represents a species in our data set. And here our primary axis of variation, PC1, accounts for 37% of our skull shape variance. And if we color code according to dentition type, what we see is that along our PC1, our principal axis, beaked species here in blue occupy a separate area of the morphospace from their non-beaked counterparts in orange. And additionally, when we calculated morphological disparity between each group, we find that beaked species exhibit significantly higher shape disparity than non-beaked species. So by contrast, if we look at habitat type, our reef and our non-reef associated species don't occupy separate areas of our morphospace. And when we calculate disparity, there is no significant difference between habitat type. And now, if we use our ancestral character reconstructions, we can look into the timing of the origin of these 2 traits. And we find that beaks have evolved separately 2 different times in tetradoniforms. And the evolutionary origin of the beak actually predates their colonization into coral reefs by roughly 15 million years. And while we find no difference in the degree of skull modularity and integration between our beaked and our non-beaked skulls, what we do find is there are substantially faster rates of evolution occurring in beaked species. So here are the skulls of an example of a beaked and a non-beaked Tetradoniform, and I've color-coded each bone according to their evolutionary rate. And you can see in the jaws of the beaked species, especially the premaxilla, maxilla, and dentary, the evolutionary rates are approximately twice as fast in beaked species compared to non-beaked species. Now, with our Bayes traits analysis, we can look at the rates of evolution across the phylogeny. So here, faster rates are in warmer colors and cooler rates are in colder colors. And I want to point out that we do find high rates of evolution on the two branches that are leading to the transitions where the beaks occur. And we also see some other interesting examples here in this filefish and the spikefish. They both have these really elongated snouts, so these may deserve a closer look in the future. So the role of integration on innovation is still a bit unclear. Typically, integration is thought to act as this constraint on phenotypic diversification. So if we have two traits, say trait A and trait B, that are highly covariate with each other, there's not a lot of wiggle room for them to expand into different areas of a morphospace compared to something that has more modular evolution. However, some more recent studies suggest that integration can actually aid in the evolution of innovation by promoting evolution along these very specific evolutionary trajectories within a constrained region of trait space. And this could play a key role in the evolution of these really maximally disparate phenotypes, like the flatfish skull. So if we circle back to our aims, we show that despite beaked species displaying really high diversity, disparity, and rapid morphological diversification, we didn't find any significant differences in patterns of modularity and integration between our beaked and our non-beaked groups. This suggests that rapid evolution of these maximally disparate phenotypes can still arise while maintaining this conserved pattern of trait integration. And integration may have less of an effect on the evolution of innovation. And finally, our results suggest that coral reef association alone may not be enough to promote large changes in morphological diversification. Compared to the species living on coral reefs, we find that species in other habitats like open oceans and rivers are just as morphologically disparate and exhibit similar rates of skull shape evolution. Additionally, beaks actually predate the colonization into coral reefs by roughly 15 million years, so it's likely that reefs did not promote the evolution of the beak. So in conclusion, beaks are a big deal, at least to Tetradoniforms. The beak evolved in the presence of highly conserved patterns of skull integration, suggesting that this integration did not limit the range of available phenotypes to Tetradoniforms. And furthermore, irrespective of habitat, beaks are associated with high rates of morphological evolution and diversification, and have allowed Tetradoniforms to diversify into novel ecological and dietary niches. All right, I want to thank you guys for your time. I want to acknowledge all of these people for their help and support on my project, especially the museum collections, without whom this work would have not been possible. And I'm happy to take questions if there's time. Thank you.
Great. We have a question in the Q&A. Says, sorry if I missed this, but were any outgroups included? Would those help put the amount of disparity and rates of evolution into context of fish diversity more generally?
So my project, we did not include any outgroups, but I am collaborating with people on a much larger project, including all of euparkerian fishes. And it's not published yet, but that will maybe put into perspective how wild and wacky Tetradoniform fishes can be, because they are very far out there in the fish morphospace. But for this study, I did not include any additional outgroups.
Okay, wonderful. Thank you, Emily.
Thank you.
Next talk in session 2 is Yuki Haba. Take it away, Yuki.
Hello. Thank you. Do you hear me? Great. All right. Hello, I'm Yuki Haba, and today I'm super excited to tell you about my PhD work with Lindy McBride at Princeton. As we populate the world, we've been changing every corner of the earth dramatically. Since tens of thousands of years ago, our agriculture and farming have engineered the landscape significantly. And as we all know, the impact has accelerated in the past hundreds of years as we've been building many complex and beautiful urban megalopolis around the globe. This global engineering has impacted all kinds of lives on the earth, from mammals to insects to plants, and led to the unprecedented era of evolution called the Anthropocene. And you can see even rats in New York City feeding on pizza, officially claiming the New Yorker position. So today, I'd like to introduce you to one of the most fascinating examples of evolution in the Anthropocene. And to do so, I'd like to take you back in time with me to a cold winter in London, 1940. In the winter of 1940-41, the Second World War was raging, and Londoners faced a sustained bombing campaign known as the Blitz. Many people sought nightly refuge in the city's underground subway system, or the Tube. However, the Londoners in the Tube found themselves attacked by yet another enemy, mosquitoes. The name of the mosquito is Culex pipiens for molestus, also known as the London Underground mosquito. And this little creature is indeed the main character of my presentation today. Scientists have studied the populations and found that there are two behaviorally divergent forms within Culex pipiens mosquito. These pesky biters found in London Underground voraciously attacked humans, could mate in confined spaces, and they were active year-round. Seeing such a mosquito perfectly adapted to man-made subterranean structures was surprising to the people, because if you compare molestus to the more common above-ground form pipiens, it mostly bites birds, requires large space to swarm to mate, and diaposes in winter. Strikingly, despite the array of behavioral and physiological differences, the two forms are morphologically identical. Even vector biologists cannot tell them apart. Although it's been almost a century since the discovery of molestus in London, the origin of the enigmatic human-biting mosquito remains contentious. So here's a question. When and where did the human-biting underground form molestus originate? The major hypothesis about the origin of molestus is what we call the in situ hypothesis, which states that below-ground molestus populations evolved from above-ground populations in situ. This suggests that the evolution happened within less than 100 years because the Tube was only 80 years old or so when the London population had been discovered. The in situ hypothesis has been fascinating academic scholars and public audience alike. Many reviews in renowned journals repeatedly cite the in situ hypothesis, hailing molestus as the best supported case consistent with the process of urban speciation. The public media, such as BBC or New York Times, wrote about molestus being the unique mosquito that lives in the London Underground. And even Bill Nye tweeted the evolution of the underground mosquito happened over the span of approximately 100 years. Evolution, am I right? So in the beginning of this project, I remember reading all of this and thought to myself, is Bill right? So we started to look for literature that unequivocally supports the hypothesis. And as we dug deep into the literature, we quickly realized that the in situ hypothesis is not well supported. First of all, the distribution of below-ground molestus is widespread, just like the distribution of the above-ground form pipiens. Instead of all below-ground molestus populations independently evolving in multiple cities in the past 100 years or so, this map made us wonder if molestus actually evolved once in a different context, and these populations were recent migrants. Furthermore, there have been records of above-ground molestus populations in the southern part of the distribution, such as Mediterranean Europe or Middle East. These southern above-ground molestus populations are ecologically and genetically similar to below-ground populations in the north, suggesting a common origin. Finally, the very first description of human-biting molestus was actually made in Egypt in 1775. So molestus has been around in Egypt at least for a couple hundred years, it seems. So at this point, I was like, Bill, it's not looking great for you. And there's no particular reason to believe that it evolved in London Tube in situ very recently. But we were actually fascinated by the inconsistency between the existing data and the popular institute evolutionary hypothesis. And we decided to conduct a comprehensive synthesis of existing data to see if we can generate a model that explains the pattern. To this end, we compiled and analyzed published genetic data at a marker gene called CQ11 for 240 populations. Here, each point represents a population, and circles and triangles represent above ground and below ground populations, respectively. X-axis is the frequency of PPN's allele, and y-axis is latitude. And molestus allele frequency is just 1 minus p. What we found was that in the northern latitude, like the UK or Sweden, above ground PPNs and below ground molestus are actually two genetically distinct entities fitting the canonical description. However, in the south, the distinction becomes blurred. Above ground populations have more and more molestus alleles, and below ground populations have more PPNs alleles, indicating that the canonical dichotomy does not hold in the south. At the southern edge, the above and below ground populations do not look different with respect to this marker locus. So our synthesis of existing data revealed a surprisingly clear genetic latitudinal gradient within populations across Europe and North Africa. That is, the stereotypical dichotomy of above ground PPNs and below ground molestus is only true in the north, but it breaks down in the south. But why would that be the case? We think it's because of the cold winter in the north. Imagine you're molestus in London, and as you breed year-round, you may try to breed above ground in the winter, but fail because it's simply too cold. This simple mechanism keeps molestus in the subterranean places where temperature or even humidity stay preferable around the year. But such selective pressure weakens as one moves to south, and in the low latitudes, such selection might not exist anymore, and the above versus below ground distinction disappears. So we started to see what was missed by the overly simple narrative of above ground PPNs and below ground molestus dichotomy. This was very exciting, but we also knew we were still far from the complete picture because our model still relies on the single locus genetic marker, and the geographic coverage of the data points are far from complete. Actually, the distinct distribution of PPNs and molestus extends far beyond Europe and North Africa to the entire globe. To really understand the full picture of the origin and evolutionary history of the man-biting mosquito, we need genomic data from a global collection of populations. So we started PipBOP, the Culex PPNs population genomics project. The goal of this project was to assemble and sequence around 1,000 genomes to understand the origin and genetic status of Culex PPN species complex. We really started this from scratch, and we created a flyer like this on the left and emailed every single Culex researcher across the world for a collaboration. We also used Twitter/X announcing PipBOP worldwide. Meanwhile, I spent several summers collecting above ground mosquitoes locally across cities in the US. I also done below ground collections on the right, sticking my head into ditches and sewages to collect larvae, which was, trust me, quite exciting. After almost 4 years of both local and global collections, we got more than 12,000 samples from 90 groups coming from all continents where it's known to occur. We then carefully chose and sequenced 840 high coverage genomes. And for the interest of time today, I'll focus on the analysis of molestus origin using the 355 genomes collected from the Western Palearctic area. But I'd be happy to talk about the global population genomes later if any of you are interested. Okay, let's just jump right into the genomic analysis result. Before showing you the full result, I will first say that using genome-wide SNPs, we found that the genomic PC1 represented the PPN's molestus axis. Our first goal here is to ask if PPN's molestus ancestry correlates with latitudes like this U shape as predicted by our analysis of one locus data. Okay, let's see what the data say. Ta-da! Our individual genomic data confirms the exact pattern we saw earlier. We were super excited to confirm the major pattern, but on top of that, the individual genomic data also revealed something that we couldn't find in our previous analysis, which is that PPNs and molestus remained genetically distinct even at the southernmost latitudes. Consistently, when we looked at the distribution across the map, we noticed that there are many above ground molestus populations sympatric with PPNs, indicating that there must be a very strong reproductive isolation between the two ecotypes. Lastly, we were excited to confirm above ground molestus in Egypt, and also found that Egypt molestus occurs without coexisting pipiens. Okay, so we revealed the complex fine-scale population structure among the Western Palearctic pipiens and molestus connected with this beautiful latitudinal climb. So the next question is, where did molestus originate? To answer this question, we built a phylogenetic tree of molestus populations from the Western Palearctic as well as other regions of the world. There are three major clades within molestus populations, and the clade that has the above-ground molestus populations from Egypt, Israel, and Greece here was basal to the other two clades, suggesting that the populations in the East Mediterranean clade are likely ancestral. The other two clades were separated not just by geography, but also by their microhabitat origin. The West Mediterranean molestus clade is mostly above ground, and the Northern molestus clade is mostly below ground. This tree provides some evidence that colonization into below ground environments likely happened more than once. We then looked at the genetic diversity of each molestus population, finding that the East Mediterranean molestus had overall very high diversity compared to the other two derived clades, consistent with the idea that they are likely ancestral. Among the populations in the putative ancestral clade, what really stood out to us were Egyptian populations because they were the most diverse populations in the clade. And remember, Egyptian populations are the only molestus that did not coexist with pipiens. This would have given molestus the opportunity to evolve without having to deal with gene flow from pipiens. So multiple lines of evidence point to Egypt as the ancestral molestus. But why Egypt? Well, if we look at the history of human cultural evolution, there are many reasons to believe that human adapted mosquitoes might have evolved in the Middle East on the order of thousands of years. The Middle East is precisely the region where the earliest agriculture and civilization first arose. And in these very dry regions, the agricultural water source must have been the only place for mosquitoes to reproduce because they have to lay eggs on the water. And for the adult mosquitoes, humans and some domesticated animals must have been the only hosts available. But at this point, all of this is just speculation. To test this hypothesis using our whole genome data, we applied cross-coalescent analysis to Egyptian molestus and Mediterranean pipiens and estimated the split time between pipiens and molestus to be 2,000 years ago, with possible range being 1,000 years ago to 10,000 years ago. This is very broad estimate, but the range nicely overlaps with the rise of agriculture and civilization in Egypt after the end of last glacial period. Also, even the most recent possible split of 1,000 years ago would be probably a bit too old for London Tube to be present. Thus, our genomic data show that the Egyptian molestus split from nearby Mediterranean pipiens thousands of years ago, coinciding with the rise of agriculture and civilization. Then, the primarily above-ground molestus might have first gradually colonized the Mediterranean region. They probably could not go up further, though, in the north for a while because as they breed year-round, they would not have survived severe winter in the north, unless there was a stable and warm shelter throughout the year. Intense urbanization in the past hundreds of years created such shelters, like basements or subways, allowing for molestus to migrate farther north into the temperate Europe, creating what we may consider today the pure below-ground molestus. So after all, Bill, doesn't look like you're right. Sorry. So the takeaway, we found that molestus, aka the London underground mosquito, likely originated in Egypt thousands of years ago and colonized urban subterranean habitats over the past hundreds of years as we built complex cities like London. I hope I have convinced you through this fascinating evolutionary story that studying mosquito evolution can help us better understand fundamental questions about the evolution in the Anthropocene. And I hope you will remember this story when you will see mosquitoes next time in New York City or London. With that, I'd like to thank my advisor, Lindy, the McBride Lab, and the funding with a special shout out to the SSC Rosemary Grant Advanced Award that made the PIP possible. Thank you. And all the collaborators. I'm happy to take any questions if there's time.
Thanks. We could take one quick one. Okay, we're going to move on. Next up, the last talk in Hamilton session 2 is Eric Iverson.
All right, thank you all for coming today. So my name is Eric Iverson. I'm from the University of Texas at Austin. I'm going to be talking today about ecological speciation and mitonuclear coadaptation on elevation gradients. I just want to thank real fast my co-authors on this talk who weren't listed because I wasn't sure what work was going to make it into this talk. But David Guevara Paza is my Peruvian collaborator who is absolutely essential for a lot of this work. Abby Criswell is an undergrad I work with who helped with coding some of the tools that I use. Ben Moran and Molly Schumer from Stanford have led some of the work that I'm going to talk about today, and my PI is Justin Havard. So ever since Humboldt, people have noticed that as you move up in elevation, you pass through these distinct life zones, and you often see related species replacing one another with elevation. One of the things we're looking at in this figure here is a series of elevational transects and birds known as tapaculos, where you can get up to 6 different species replacing each other with elevation in this sort of staircase pattern, with different groups of them existing at different places in the Andes. And we've known for a while that genetic differentiation often comes with living at different elevations. We might have different species, but we also might just have different populations. And we often see that species where even the nuclear genome is similar across elevations are showing differentiation in the mitochondrial genome. So if you look at these rufous-collared sparrows here, the nuclear chromosomes are represented as rectangles, the mitochondrial ones as circles, and you can see that while the nuclear genome is similar, those mitochondrial genomes appear to be diverging across elevation. And this relates to a lot of work that has happened in both humans and other animals, showing that mitochondrial proteins and mitochondrial processes are really important for adaptation to different elevations due to their role in metabolism and regulating adaptation to different temperatures and oxygen levels. So what's going on inside the mitochondria? Well, basically in the process of oxidative phosphorylation, we are creating usable energy from inputs that come from our diet. And we are pumping protons across a gradient, and then those protons are released back to fall through a protein that can basically spin and phosphorylate ADP. We can have uncoupling proteins, which are going to increase the rate of flow through that membrane and allow us to generate heat. That's something we do as endotherms. This process also involves the release of reactive oxygen species, which can have a role in cell damage, apoptosis, and aging. And so we can see how different environmental factors such as our diet or primary productivity, the temperature at which an organism lives, or the availability of oxygen, UV radiation, and other stressors are going to impact this process and cause selection upon mitochondrial proteins. Now, these mitochondrial proteins don't act in isolation. They have really intimate conformational arrangements with nuclear proteins. So what we see in this diagram is that those respiratory proteins involved in oxidative phosphorylation, well, the green components here are encoded by the mitochondrial genome, while the yellow ones are encoded by the nuclear genome. And all those orange and red places are residues where proteins from those 2 different genomes are coming together and interacting. And because proteins from these 2 genomes evolve at different rates and under different circumstances in response to different pressures, we can have a situation where there might be a poor fit between these proteins. So if we imagine under normal function, these proteins are fitting one into another like a glove, if there's a mutation that gets fixed in one genome or another for some reason, it may cause a change in conformation that results in a gap here where those 2 proteins are not interacting as well as they could be. But then we could have a compensatory mutation that gets selected for, and when that's fixed, we restore that function by basically bringing back the conformational fit that's so important to these proteins. Now, a lot of work has demonstrated that interactions between the mitochondrial and nuclear genome, what we call mitonuclear interactions, are really important for determining properties like metabolic rate, and they also interact with the environment. So we talk about G by G by E interactions, which basically influence fitness. So in the example of this flower beetle here, work has shown that different mitochondrial lines, which are represented by the different shapes on the graph and the lines connecting them, if you place those on different nuclear genetic backgrounds, which are along the x-axis, then what you see are different metabolic rates. So every combination of mitochondria and nuclear background results in a different metabolic rate. And also between the top and the lower panel, we have different temperatures, and you can see that those relationships get scrambled depending on which temperature you run them at. So that's illustrating a G by G by E interaction. And a lot of work has illustrated how these mitonuclear interactions are really important for reproductive isolation. So some work I was involved with in collaboration with Stanford University, we showed that in this hybrid system between 2 swordtail fishes from Mexico, that live at different elevations and where they form hybrids at intermediate elevations, there's actually a lethal incompatibility between certain mitochondrial and nuclear genes that enforces reproductive isolation. So based on looking at ancestry in some populations, we identified specific loci that appear to be outliers where you basically never see certain genetic combinations in that population, because if you're mismatched there, if you have the wrong mitonuclear combination, you're not going to survive to adulthood. And we've isolated that down to a specific location inside of complex 1 of the mitochondrial genome. And through doing functional analyses of the mitochondria, where we isolate those mitochondria, place them into a chamber, and we watch oxygen go down over time, which is the blue line represented here, and we add different substrates and inhibitors, we can see how the flux of oxygen changes over time. So that's the red line going up and down in relation to what we've added. And by doing this, looking at these isolated mitochondria and their respiration rate in a chamber, we can show that in F1 hybrids of these 2 parental species of swordtail fishes, they have a lower efficiency of complex 1, which is what we would expect if they were displaying an incompatibility between the mitochondrial and nuclear genomes. And in other mitochondrial properties that you can see on the right there, there's no difference, or they're intermediate like we would expect them to be. So this is evidence for the importance of these kinds of incompatibilities in driving reproductive isolation between species. Now, some of this work led me to wonder whether, given how important mitochondria are for adapting to the environment, and given the importance of mitonuclear interactions in causing reproductive isolation, whether we might be able to use this information in a conservation framework and mimic a natural process whereby it seems that often in history, mitochondrial genomes have introgressed from one species into another. And when they've done that to overcome those mitonuclear incompatibilities, we've often seen the co-introgression of particular nuclear alleles that are important along with them. So I propose this idea of conservation mitonuclear replacement, where we could facilitate adaptation by basically going along the pathway that we would see below the dashed line here, where we use a combination of editing those key NMT loci, that's what we call those nuclear genes that interact with the mitochondrial genome, and moving mitochondria from one species to another in a way that might be adaptive in a future scenario, for instance, under climate change. So one way this might help when we're talking about adaptation to elevation is as species are forced to move upward in elevation, some of them might be squeezed out by species moving up from below them, like we see in this first scenario here. So if you imagine moving from left to right in that temperate biome there, those species which are represented by a circular mitochondrial genome and those 2 nuclear chromosomes are going to be forced upward in elevation. If they can't adapt to a lack of oxygen, they might go extinct. But we might be able to take some adaptive variation from a lower latitude, from a subtropical area, bring that mitochondrial genome and those specific nuclear genes that are important into that population and allow them to move upward in elevation, or perhaps allow them to persist at an elevation where it's gotten hotter. So there's a variety of applications for this, and if you want to learn more about it, the QR code there will take you to that paper. So we wanted to test some of these ideas in the field and basically see if we could detect adaptive differentiation in mitochondrial function across elevations. So to do that, we went with this group of birds called tapaculos that are found across the Andes in South America. And across the phylogeny of the group on the left, you can see those branches are color-coded based on the elevation at which those birds live at. So there's a wide variety of elevations represented here. And we went to one specific area of southeastern Peru where we have 4 different species with increasing elevation. They're in this sort of 2 pairs of sister species. These are the species, and we see across the transect how they segregate with elevation, going from lower to higher across Peru. They appear to be good species that are reproductively isolated. Looking at their calls here, we can see that they have very different calls based on trill ratio, so they probably recognize each other as different species and don't interbreed anymore. And so what we wanted to do was, with that increasing elevation across these 4 species, measure their basal metabolic rate and also the heat production of their mitochondria to see if we could detect adaptation to elevation. So I worked with an awesome team of Peruvian and American collaborators to catch these birds all across the area surrounding Manu National Park. And one thing we did is to basically, in a metabolic chamber, lower the temperature overnight and see how the whole organismal metabolic rate changes. And because these are endotherms, we expect that as it gets colder, moving from left to right over the course of the night, we're going to see that their consumption of oxygen is going to increase. And at a certain point, we're going to see a break in that graph. And we can use AIC to determine where that break point should be. And we call that the lower critical temperature, which is where these endotherms start to increase their respiration. So looking across these 4 species, we see a decrease in those lower critical temperatures. So we've got our low elevation species, then a medium elevation species, and then 2 high elevation species. And we can see that the average LCT is represented with the red dot there. That is going down as we move up in elevation, which is suggesting some adaptive differentiation in the metabolic system of these birds. Now, what about the mitochondrial component to that? Well, again, I mentioned how mitochondria can generate heat through the uncoupling of that mitochondrial membrane, allowing protons to pass and dissipate their energy as heat. So we brought a mitochondrial respirometer to the field. This is one of our undergrads using that in Peru. And what we found was that in both the brain and the liver tissue, mitochondria isolated from these tissues don't show differences in their capacity for heat production. This is what we would call leak respiration. So we're not seeing those adaptive differences there. So if I bring back the whole organismal phenotype, which is that lower critical temperature on the right, and compare it with the leak respiration, we can see how we may be having different things going on at the level of the mitochondria versus the level of the whole organism. So this prompts the question of whether there is adaptation in the mitochondria. Maybe there is adaptation that's not represented just in the leak phenotype. Maybe there are differences in the number of mitochondria that could explain differences in the whole organism metabolic rate. So we have some work we want to do, basically looking at mitochondrial copy number and starting to analyze the sequences of the mitochondrial genomes from the individuals that we captured there. So zooming out a little bit, this prompts the question of the relative role of adaptation in the mitochondrial genome overall, relative to other processes that might be explaining some of the signatures people have found over the years in the mitochondrial genome. For instance, the DNDS value, which is the ratio of non-synonymous to synonymous substitutions, is often claimed to be higher. And in all these species, high elevation species have shown some evidence from DNDS ratios for adaptation in the mitochondrial genome. So we wanted to ask, actually, what is the ratio, or what is the relationship between adaptive and neutral processes in the mitochondrial genome overall? And essentially, we need to have a framework here where we can differentiate positive selection from purifying selection. And so we want to work with both the DNDS value and also this value called the K value. And some work that's come out of my lab, we've sort of worked up a technique where by looking at both of these values in a phylogenetic framework, we can begin to isolate whether evolution is neutral or whether we're seeing actually intensified positive selection versus relaxed selection or other things that could be going on that are driving DNDS values to change. So I'm going to skip over the results here a little bit, but just keeping in mind that we need those two values, our DNDS value and our K value, that tells us whether selection is relaxed or not. We worked in this 3 taxa framework where basically we're looking for a high elevation species on the left, a low elevation sister species, and then a low elevation outgroup, and calculating DNDS values along those 3 species trees, and then analyzing those 3 species trees across a variety of different taxa. And so this work is on bioRxiv now if you want to check it out. But essentially what we found is that the high elevation species does indeed tend to show a higher DNDS ratio, so more non-synonymous substitutions in the mitochondrial genome in all of these groups except for the fishes. And we see some evidence that rather than being related to absolute elevation, actually, some more interesting ecological properties that shake out are the ratio of range sizes between species. So if you have a high elevation species that has a much smaller population relative, or a much smaller geographic range size relative to its low elevation sister species, then it's going to show a higher DNDS ratio relatively. And so we looked at basically the fact that species that live higher in elevation tend to have smaller ranges. Those with smaller ranges tend to show that higher DNDS ratio, and also some evidence from K values that selection was more relaxed. And so it looks like rather than being adaptive, what may be going on here is that high elevation species tend to have smaller population sizes, and thus they're going to show those signatures of higher DNDS ratios. And so I think I'm running out of time. I'll just say that we also looked at this in a larger phylogenetic framework, and we showed that in a bunch of different groups, DNDS values increase with elevation, and those K values decrease with elevation, as we would expect. So this, again, points to a pattern of high elevation species having smaller ranges, smaller effective population sizes. That increases genetic drift, leads to higher DNDS ratios. So it's not necessarily an adaptive process. Rather, they shake out here on this graph. And so that just contributes basically to my conclusions that both neutral and adaptive forces may be driving evolutionary divergence between species at different elevations. Both mitochondrial and mitonuclear differentiation can contribute to reproductive isolation via mitonuclear incompatibilities, and these loci are key loci that we should be looking at in conservation genetics for their role in adaptation and reproductive isolation. So with that, I want to acknowledge my funders, my collaborators, a couple field stations that have really helped me out, and a bunch of undergrads, lab members, and people from other universities who have helped with all of this work, which is just a ton of work. So thank you all so much for this, and I'm happy to take any questions.
Thank you, Eric. It is 3:45, so we've reached the end of our session, and I want to thank all of the speakers for a host of amazing talks. Truly, thank you. I'll see you back for session 3.